Welcome, class. To I am Alberto Villarengo, and today we are going to discuss signal transduction and how it regulates the cell cycle. Now, show of hands, who here knows what who here has prior knowledge of signal transduction and how it works? No one? Okay. Well, don't worry, you'll get a nice good brush up with this. Video link will be posted later once this one session is complete, so you all can visit it again, this lesson again. Now, let's talk about let's introduce this a bit. Living cells, whether they be prokaryote, eukaryote, in a unicellular or in a multicellular organism, need a means to sense and respond to the environmental stimuli in their habitat. They do this via with a chemical process called signal transduction. Cells use this to respond to the habitat around them, changes in the habitat around them, and act accordingly. They can also use this to respond to trigger and respond to changes within themselves too. By understanding this process, we and the various functions it brings about, we humans have managed to greatly advance medical science and genetic engineering and whatnot to in great strides. And doing this, we still continue to make new applications and improve existing ones. Before we conclude this lesson, we will go over different topics, which features of signal transduction, what makes signal transduction happen the way it does. We'll go over the cell growth cycle and the different both in general and in detail. We'll go over how signals affect the cells, with the mechanism behind it and different examples on how it happens. Then we'll get to cell growth signals, like the signals specific for cell growth, and also apoptosis, which is a programmed cell death. We'll get to that when we get to that. And we'll go over applications of how we humans can apply this knowledge to various sciences and technology. All right. Everyone on board so far? All right. Now, who here... Well, we already said here, but we already established that that you guys aren't too familiar with cell signals. But who here has an idea? Just shout out a general idea. Okay, I gave you a chance. We'll just go through it here. Think on this. Signal transduction, there's different types, and all these types have different features. The features in question, we have specificity. Each receptor in a cell will respond to a specific signal protein. Okay, like there's a receptor for one protein will not necessarily fit another protein in it, or respond to another protein or molecule, whatever is being used to signal. Amplification is another feature. Every time a receptor is triggered, what it does is it cascades, it triggers sends out a whole bunch of further signals that trigger even more receptors, cascading until it eventually triggers the desired effect or the intended effect function in the cell. Modularity is another cool feature. Some signal proteins, they can have multiple binding points with different affinities for that binding, which can not only change how long they stick in a receptor or not, and also cause receptors can also modulate too, act in reverse depending upon whether they're phosphorylating or not. Phosphorylating being the breakdown of phosphorus and phosphate bonds on molecules. Another cool feature of receptors is desensitization. You see, receptors, when they're active for a long enough time, they trigger that cascade. And when that cascade goes on long enough, the signal proteins from that further signal proteins from that cascade will create a feedback reaction that inhibits the receptor and keeps it from and stops it from producing that cascade any further. That overstimulation, in other words, receptors, as they keep going on, will desensitize, turn themselves off, start to turn themselves off. That's like why some people have like issues with like allergy medications stop working after a while. The receptors that trigger that respond to antihistamines start to become desensitized, and hence the antihistamines start losing their effect. Another cool thing with receptors is integration. You see, you can have two receptors with opposite effects, basically, and they'll still be active. If both are activated, though, one will have a net one here, whichever one is greater, they'll have a net effect, meaning you basically add. Let's say you have a receptor that has an effect of 3 minus, and another receptor that effect has of 2 plus. Yeah, the net effect. You add them together, they integrate, and you get a net effect of 1 minus. All right. If you could put the effect into numbers, that's basically how it works. Another cool thing is receptors go along different pathways. Although the cascade works basically by floating about in the cytoplasm and to trigger, eventually bumping around and triggering the response, which we'll get in more detail on that later. There's a certain pathway thanks to cell proteins and enzymes in the molecule, enzyme molecules in the cell, that keep the receptors further signal cascade from straying too far and hitting on and inhibiting other receptors. You see, basically, yeah, you have the message-destroying enzymes. They form around along the cytoskeleton, along the signal pathway, and just sort of keep the cascade within the right area. Now, these are all different features of each and every type of, generally every type of signal receptor and signal transduction, but there are different types of signal transduction too. G proteins are special types of receptors that occur in the plasma membrane, the lipid bilayer of the cell membrane, and they take in the extracellular environment, the environment outside the cell basically has receptor signals in it that once they float about and binds to the G proteins then send signals into the further signals, the cascade, into the cell. And in doing that allows the cell to respond to external stimuli. There's also enzyme receptors in the cell. They're known for handling things like intracellular growth. They allow the cell to grow in itself, as well as grow different appendages, like allow nerves to grow their long axons and their long axon strands to reach out and branch. Another cool thing, another type of signal transduction is gated ion channels. These are pretty specific to nerves, because these allow neurons to neurons can conduct electrical signals, but within themselves, but have to transition to chemical signals to get to the next neuron and back, and then back to electrical signal. Gated ion channels allow nerves to send their electrical signals out of them as like chemical messages, which then get received by another nerve and are then converted back into electrical, and so on. And then when it comes to multicellular organisms that need with specialized cells, like us humans, other mammals, fish, even cnidarians, like jellies and anemones. You have nuclear receptors that lipid-soluble steroids will bind to. See, lipid-soluble steroids, they can float past. They don't have to be taken in through a cell, an external receptor. They can just float through the lipid bilayer, being lipid-soluble. And they get into the nucleus, bind to the nuclear receptors on the chromosomes, on the chromatin, and change gene expression. They activate genes on the DNA to be more expressed or not. So these are some signals can get pretty complex, and there are a lot of different types. And we'll get to that, and now we're going to go into the cell growth cycle. Later, in a few slides, we'll get to how signals affect the cell growth cycle. It'll make sense. On this cell growth cycle, you have two main phases, which are broken down to further phases. The inner phase is the first big phase. This is where the cell is growing, basically, before it starts to divide. G1, the cell is growing. It's generally accumulating nutrients and energy and growing in size. Once it grows big enough, the S phase triggers, where the DNA in the cell starts to replicate. The chromosomes will copy and form into little X shapes, to kind of like X or knot shapes, in order to at the ready for when the cell eventually splits. And then G2, the cell's growing even more in size. And this time, it's not just growing in size and replicating DNA, but it's also replicating its other structures, organelles, membranes, mitochondria, and ribosomes, and so on. When you get to the meiotic phase, and then once that happens long enough, the meiotic phase triggers. Meiotic phase is basically mitosis and cytokinesis. Mitosis, the cell happens when the cell starts splitting into two, is when the cell begins to start its splitting. The nucleus is splitting, the external membrane starting just first, and then by the end, the external membrane is starting to split. Cytokinesis occurs when that membrane when the external membrane is starting to completely split, eventually pulling apart into two daughter cells, which then repeat the cycle. Okay, the daughter cells then grow, enter in G1 and S, G2, and so on. The cell cycle repeats and the cells multiply. Now, here the cells get into a little more detail. You start with the G1 interphase, okay? In the G1 interphase of interphase, you can see the cell is growing, okay? Not much really change is happening inside. But when you get to S phase, you can see that the inside the nucleus, this little purple spot here, you can see the DNA strands marked by the red and blue chromosomes are starting to become more. They're replicating. And then you'll see here in G2 of interphase, the chromosomes are replicated and neatly organized. And you also see that it's kind of hard to see in this picture, but you'll see that they look like kind of little suns with strands between them. The strands are starting to push the suns apart. That represents in this image, that represents the organelles replicating and pulling apart as the cell grows bigger. And we get into the meiotic phase, starting with prophase, the cells where the nucleus, prophase here, okay? Meiotic phase, the prophase, the organelles are replicated sufficiently, and the centrioles, which are like little stiff little strands, fingers that push the cell apart, are starting to push the cell apart. When you get the pro-metaphase and metaphase, is when the nucleus is basically starts to dissolve. In pro-metaphase, the nucleus starts of mitosis. The nucleus starts to dissolve, and in doing so, the centrioles can start grabbing onto the chromosomes, which you see here in metaphase, which then leads to anaphase, where the chromosomes are pulled apart, where the replicate chromosomes are pulled apart by the centrioles, so that the two daughter cells can then start forming a new nucleus around each around the replicates, leading to telophase and cytokinesis, which is behind my head here, where the cell starts to split, new nucleus is formed, and the DNA is organized inside as such. And each new daughter cell, as I said earlier, repeats into G1 and S and G2 mitosis and so on, multiplying the cycle. Going back to signals here. Signals affect cells through a thing called through a cascading, but that's a cascade is initially triggered by essentially chance. You see, just like when molecules bond to things, they float about randomly, and they just have to hit the right orientation at the strike each other at the right orientation to bond or react. Signaling happens the same way. However, cells ensure that the signals happen by producing a whole bunch of signal molecules and a whole bunch of receptors to receive those signal molecules, so that the bumping is guaranteed to occur, so that the correct binding is guaranteed to occur, regardless of the randomness of the bumping and so on. And orientation, randomness of the orientation and the floating about of the signal molecules. After the receptors get triggered, and after triggering, they release a cascade that releases even more signal molecules onto more receptors that in turn eventually activate the biochemical change. And as you can see by the diagram here, each part of the cascade, once it releases an increasing amount. The first step, one molecule manages to bind to a receptor. That receptor triggers a cascade that releases 10 squared or 100 molecules, which releases another 100 molecules, and then into another bunch of receptors, and then so on, increasing to, like, let's see, 10 times 10 is 100 times another 10 is 1,000 to 10,000 molecules, and 10,000, then 100,000, and so on until the that cascade eventually reaches the final receptor, triggering the final desired biological effect. Now, all these cell signals will affect can happen in different ways. You have ionotrophic receptors. They can these cascade signals generally how things happen. However, you have like different types of cell signals, okay, for different biological functions. Like ionotrophic receptors are interesting. They're G protein linked. They relate to the G proteins, but they have an ability to not only just send the cascade, but they just both sense the signal and trigger an effect of their own, in addition to sending the cascade. They can be either voltage or ligand gated. Ligand gated being the response to a specific molecular ligand, or voltage gated being the response to a change in electrical potential. You have your endocytic receptors. An example of these, they're found on white blood cells and like neutrophils and macrophages, and being located on the surface of these white blood cells, they're there for antibodies to bind to pathogens, and then the white blood cells can recognize those antibody-bound pathogens and engulf them, binding to the phagocytes and so on. And then you have, this is where when we start to get into cell growth, you have growth factor receptors. You see, one of the things cells need to grow is energy, which they get by making ATP from sugar. Insulin receptors allow cells to give cells insulin is a signal protein that, when bound to the right receptor, will open up a cell to receive sugar molecules and energy. Basically, insulin receptors, when they're triggered, they dictate calorie entry. It allows cells to give the cells the energy to do their functions and continue in the cell cycle. You also have cells like receptor tyrosine kinases. These cells, like in neurons, they allow for epidermal growths, like neuron branches and such, or other additional structures for specialized cell features. Now, here are some examples. Here are some ways that how cells, some signals that are specific to cell growth. CDK1, that's cyclin-dependent kinase 1. It's an enzyme-based receptor that regulates the transition from interphase where the cell's growing and replicating its DNA and replicating its organelles and growing some more. It regulates that transition from that into the meiotic phase where the cell starts to make the centrioles that branch out and pull the chromosomes apart, the replicate chromosomes apart. One of the things that CDK1 does, it features, is it also condenses the chromosomes into their compact chromatid rods. It does that during the interphase. When the chromosome rods start to replicate chromosome rods compact, form into duplicate chromatids, like X's. Then, as meiotic phase is going on, when meiotic phase starts, mitosis starts, the anaphase promoting complex, or APC, kicks in. This keeps mitosis going and promotes it from going into cytokinesis. It allows the separase enzymes, it triggers the separase enzymes on the centrioles to grab the chromatids and separate them, cleaving them, the proteins responsible for holding them together, and pulling them apart into each daughter cell. In doing so, it also deactivates the cyclin-dependent kinase as part of the cascade that it produces. Otherwise, the cyclin-dependent kinase would keep the cells in a state where the chromosomes, otherwise the chromosomes couldn't be pulled apart, and then they wouldn't uncondense once the cell's completed. Because once the cells separate, the chromatids need to uncondense so that the DNA can be replicated again in the next cell cycle, in the continuing cell cycle. If that didn't happen, you'd get some of the cell would usually trigger it, and leading to something called apoptosis. Apoptosis, it's something that cells do when they recognize something's wrong with their cell cycle. You see, when a cell recognizes the cell has other signals, like checkpoint signals around it or throughout its cell cycle, and if something bad happens, usually the cell triggers apoptosis, where it neatly kills itself so it can be digested by a neighboring cell. Kind of unlike something called necrosis, where the cells sort of just break apart and spill over into a cascade of death for the neighboring cells. Apoptosis is a neat shriveling of the cell, where it eventually shrivels and falls apart just enough for the neighboring cells to engulf it and digest it without any further a cascade of cell death, which necrosis has. When this apoptosis occurs, when the by cells responding to malformed cell growth and triggering capsases. Important to note, the proteolytic cascade caused by capsases, proteolytic meaning the breakdown of proteins, is irreversible. I mean, sure, there's a few select proteins that can be undenatured, but denaturation of proteins is generally irreversible, and unless it's like digested and reproduced in another cell, but that's basically not denaturing, that's digestion and remaking. Irreversible proteins, apoptosis is irreversible, especially once it reaches past a critical point, but it occurs cleanly and quickly for each cell, keeping further damage from happening to other cells that in turn digest the shriveled up suicidal cells, I'm going to call it. Now, all this stuff has various applications in science and technology, modern science and technology. And medicine, cellular function, as we discussed with like insulin earlier, cellular functions are dictated by signal molecules. Okay. By studying these, we humans have devised drugs containing synthesized signal molecules that can stimulate functions that certain individuals with certain disorders are lacking in these functions. They can also inhibit these. We can also have ways to inhibit cellular receptors or overwhelm them, inhibit them with blockages or feedback loops. With blockages, feedback loops, or net effects in order to stop cells that are over functioning, cell receptors that are over triggering. Like some autoimmune diseases like rheumatoid arthritis, the immune system attacks the joints, and the immune system is overstimulated. And so treatments for rheumatoid arthritis could include things like you say by creating stimulating other receptors to stop the resulting inflammation. You just with enough to overwhelm, create a net effect that stops the inflammation, but it won't stop the receptors triggering the inflammation, but it will stop the inflammation itself with a net effect by triggering other receptors that calm inflammation. Examples of medicines that trigger or block suppressors include steroids. Like if you want to trigger receptors on your certain genes to trigger genes for protein growth or anti-inflammatories, that works. Immunosuppressants are an example of receptors. They like antihistamines, as I mentioned earlier. That's immunosuppressants. And cancer drugs, like you can sometimes cancer, you have drugs that inhibit receptors that when malfunction cause cancer. Another interesting thing for cellular receptors is absolute genetic engineering. You see, cells, as like with steroids, for example, cells use signals to control protein synthesis, and gene expression by making our own, as an example. But we also have ways to make imitation signals that allow cells to accept foreign DNA and splice it into the genome of an organism still developing. This allows us to create biosynthesized commodities in larger amounts, like xanthan gum, which is like we genetically engineer bacteria to produce gum products that we then harvest off the bacteria as like a fine powder. Then you have corn syrup. We splice genes into corn to make corn produce more starch, and with that we can make more corn syrup and certain oils too, and such. Like certain oils can be made with genetic engineering that encourage plants or other life forms to produce oils, and we can do this by introducing enzymes and signals to receptors, allowing cells to introduce foreign DNA and splice it into their genome. In conclusion, you see, signal transduction is a critical aspect of living cells. We humans have studied it enough to figure out how to synthetically manipulate it for various purposes as it dictates and runs the functions that our cells and other cells and all sorts of other cells perform, and they perform it through molecular amplifying cascade. Not to mention complex organisms with like nerves and muscles and other specialized cells contain signal pathways for other features like nerve signaling, apoptosis, and producing and immunosuppressants and immune system responses. Long story short, without cell signals, organic life we now live as we know it could not exist. For further references, for further reading, these here are a list of the references I used in this presentation. You are welcome to take a screenshot of this and look up these yourself if you have any further reading. I will assign homework via email.